Our next speaker is Alissa Fogg. Alissa is a stewardship ecologist in the Working Lands Group at Point Blue Conservation Science. Alissa works with farmers, ranchers, tribes, and land stewards across the state to facilitate wildlife habitat restoration practices and to study the patterns between those practices and biodiversity indicators. When not working, Alissa enjoys spending time with her family and dragging them along on birding and bicycling excursions around Humboldt Bay.
So my name's Alissa Fogg. Thanks for the introduction. And I'm presenting on novel climate change adaptation strategies for conserving drought-adapted blue oak genotypes in California. And I really need to acknowledge my main collaborator. This is Dr. Blair McLaughlin at UC Santa Cruz. She's an expert on California oak conservation. She's really spearheaded this effort. And just as a disclaimer, I'm like a trained wildlife biologist. I'm not a plant geneticist. So as I go through my talk, you may have questions that probably should be directed to Blair. And I do have her contact info at the end of the presentation. So you can reach out to her for any follow-ups. Okay, let's see. So the recent mega drought in California, this is the one from 2013 to 2015, brought about huge changes in forest and woodland systems across the state. And in the southern part of the Blue Oak Range, we really saw a lot of massive mortality of oaks. These oaks were over 300 years old. And this photo here, this is taken in one of the driest parts of the Blue Oak Range in San Benito County, west of Fresno. And blue oaks, they're the most drought-adapted, thought to be, oak species in California. Thus, seeing these blue oaks that had lived through these past droughts perish was a wake-up call for ecologists that we might be losing the trailing edge genotypes from the southern and drier extent of their range. Blue oaks, they're endemic to California. They have a narrow range that follows a bathtub ring pattern around the Central Valley. And this dieback occurred in the Tehachapi Range and along the Sierra foothills and in the inner Coast Range of San Benito County. Blue oaks, they're a foundational species in California oak woodlands, and they provide habitat for over 400 wildlife and countless insect species. They're highly valued and stewarded for millennium by indigenous communities who continue to this day to care for our oaks. And oaks, they provide beauty and shade, and their leaf drop really contributes to soil fertility for rangelands. And they confer many benefits for a lot of the privately owned ranches that run cattle on these lands. So why are we so concerned about losing blue oaks and their trailing edge? I mean, just for, losing oaks, just direct loss of any individual trees, it's going to shift their ecosystem away from a woodland and towards a grassland or desert. And this is already happening on the west side of the San Joaquin Valley. But more importantly, we were concerned about losing a part of the genome that is likely most adapted to those extreme climate conditions. So if you picture, you have an area of oak mortality, but you have the ones that survived. And these are the ones that may have those genetic adaptations to future drier and hotter conditions. So we started to think about how we could conserve these climate adaptations and what our options were. So one option is assisted gene flow, and this is the intentional movement of organisms between populations within a species' current distribution. And this is different from assisted species migration, where one may move a species to an entirely new part of outside of its range. So because the climate is changing so quickly, individuals and populations may not have time to adapt, especially with species that do not produce seed or viable young for many years. So likely candidates for assisted gene flow include those species with long generation times where it might take 30 or 50 years to produce viable seed, also reproductively isolated populations where they may occur in islands, and short dispersal distances. So for example, acorns, they rely on birds and squirrels to a lesser extent. Notably, California scrub jays cache their seeds some distance from the adult oaks, but most acorns just fall within the drip line of the parent tree. and also sessile species. So these are those that cannot move on their own, such as plants. So there's lots of existing concerns around assisted gene flow. This is not an exhaustive list, but some concerns are you could introduce maladaptive traits such as to diseases or mismatched flowering or leaf out timing. You could also have outbreeding depression where you are mixing genes from very different populations and those combinations end up being incompatible. You could have an uncertain climate future. So we know we're getting warmer. We are already much warmer. But trends on precipitation patterns are less clear. We could be moving these genotypes to locations that are too wet. But something we've heard time again is we could be risking a loss of purity of genetic lineage and the relationship that a plant has with its environment and its community. So a particular blue oak that's 300 years old and producing acorns likely evolved with indigenous stewardship, and moving those acorns to a new area will likely not confer those same benefits. Even removing their acorns from an existing site takes them away from the animals and organisms that rely on them. So how can we approach protecting these blue oak genotypes also and kind of acknowledging all these different concerns? Let's see. Uh-oh, my slides are not advancing. Hold on a minute. Uh-oh. Okay, there we go. Sorry about that. So one option is seed banking, and the photo on the right, this is a photo of the, I'm going to pronounce this wrong, but I think it's the Svalbard Global Seed Vault in Norway, and it produces long-term storage of duplicates of seeds from around the world, notably a lot of the food crops. However, acorns have recalcitrant seeds, meaning they can't tolerate conventional seed storage at freezing temperatures and desiccation required for that long-term storage. And there's other species like maples, walnuts, and buckeyes. They have the same seed type, and even coffee, which is like a little bit frightening, has the same seed type. So we came up with an idea to install climate-adaptive participatory field gene banks for drought-adapted blue oak genotypes. And Point Blue, who I work for, I'm in their Working Lands program. We have relationships with ranchers, land trusts, tribes, land stewards throughout oak woodlands in the state. And much of the blue oak woodlands are privately owned. They are working agricultural landscapes, and they generally don't have public access. And Point Blue has a partner biologist program where we have staff in counties across the state building relationships with landowners who are interested in stewarding their land for ecological benefit. And with these landowners, their land is usually held in some type of conservation status, such as conservation easements. So not having any intention of developing that land. So this trust and land access we've developed through our partner biologist program led us to establish paired plantings of these drought-adapted trailing edge xeric, meaning dry edge, acorns gathered at the southern extent of the blue oak range, and they're paired planting with locally gathered acorns. So we planted acorns in these seed baskets. They're made of hardware cloth and livestock exclosure cages that confer protection from gophers, ground squirrels, voles, birds, and cattle and deer browse. We didn't include any supplemental irrigation or any soil amendments other than we applied some mulch from nearby oak trees to inoculate the soil with hopefully with symbiotic fungi. And we did use weed suppression mats to also help retain soil moisture. And we planted 2 acorns in each seed basket. And if both acorns sprouted into seedlings, we thinned the smaller seedling. So this figure on the right shows in red where the xeric edge acorns were gathered in the southern part of the blue oak range. And then the blue triangles overlaid on green polygons, which represents projected refugia for blue oak, shows where we establish those gene bank locations. And we have sites in 8 counties across 12 ranches. Encompassing, these are 76 paired plantings, and we planted over 300 acorns. And planting occurred in winter 2018-19 and also 1920. So the table, the bottom table shows the average precipitation and temperature between the xeric edge and the planting sites. And you can see there's a difference in over 50 centimeters of precipitation in 2019, our first winter of planting. And although the difference in amount of precipitation was similar during the second winter of planting. So we published our first 2 years of monitoring data and restoration ecology a few years ago, and we were really interested in seedling survival compared to those different growing seasons. So the top figure on the right shows that the seedlings from locally gathered acorns had overall higher survival during the 2019 growing season. It was 86% compared to 57% for the drought-tolerant, the xeric edge seedlings. However, that gap narrowed in 2020, where survival stayed constant for the drought-tolerant seedlings, but it fell down to 71% for the local seedlings, possibly indicating less adaptive capacity to handle drought. And the second figure shows the difference in seedling height. So where the local acorns grew taller seedlings compared to the drought-tolerant, which could indicate a trade-off between growth and water efficiency, or the drought-tolerant seedlings, they may have put more energy into their roots. So with this, we decided a living seed bank is a viable strategy for preserving genetic diversity, but further study was really warranted for understanding, well, are we planting like the acorns that actually have those genetic adaptations? Oh, we were also interested in how disease and herbivory may differ between the local and xeric edge seedlings. These are, again, results from the 2021 manuscript. You can see on the left box plot, on the y-axis, we have leaves infected with brown disease spots, and we had a higher rate amount of leaves infected with the local seedlings compared to the xeric edge. And that pattern also held for herbivory, percent of leaves that were affected by herbivory. So fast forward a few years, we've had several outreach events, we've held planting workshops. We were also successful in securing a USDA NIFA grant to investigate the genetic component of drought adaptation. And ensuring that helping to ensure that acorns we are collecting for future gene bank would have that adaptations to future drought. So we completed monitoring at our field gene bank sites again in 2022 and 2024. And we also expanded, we're calling this the basic field gene bank, to 2 more sites. I just planted one on Friday in Shasta County, actually. So our results, these are just from monitoring we did in July of 2024, comparing the 2020 monitoring season to 2024. So these seedlings are 5 and 6 years old, show that survival has decreased for the local seedlings by 35%, compared to the drought-tolerant seedling survival has decreased by 25%. And we're actually now down to 33 of the local seedlings and 22 of the drought tolerant. We've had a couple gene bank locations actually burn in wildfires, and we had another one that inadvertently got burned by a prescribed fire. So it just kind of makes it all the more important to get more of these established. We've seen that the local seedlings have held their taller edge. So this is the figure on the right for seedling height. For those that were initially planted in that wetter year in 2019, but heights are similar between the local and drought-adapted seedlings for the ones established during the drier year. For disease and herbivory, we didn't see any sign of powdery mildew and very few brown disease spots during surveys this year. For herbivory, the percentage of the live leaves that had signs of herbivory, we did continue to see a higher amount in the local seedlings. So our next steps include genetic analysis and identifying traits that could be used to increase population resilience. We also want to identify those blue oak populations that have drought-adapted traits. We want to ensure they are protected and that they can continue to be stewarded. We also want to create best practices for assisted gene flow translocations. We actually just held a convening this fall with quite a few just oak woodland folks and tribes that are stewarding oak woodlands, just to ensure if you're collecting acorns, you only collect what you need, you have permission to collect, and acorns are handled appropriately and with respect. And we'd like to expand the field gene bank to more sites using acorns. We're looking for that funding. Right now, we're looking for potential landowners as well. And we're also going to continue monitoring and outreach to a diversity of land stewards and just keep this conversation going. It's kind of like, I think for some in the restoration community, it's a tough one to really think through, but I think it's important. And with that, I think I got through my slides pretty quickly. I'd just like to thank all the ranchers and land trusts and stewards who generously let us collect acorns. Also the Erica Zavaleta Lab at UC Santa Cruz. They are the ones who are really behind the genetic analysis and just understanding the mechanisms of drought adaptation. And I'd also efforts and just their relationships with landowners and building that trust, and our funders as well. And if I have time for any questions, I'd be happy to take them.
Thank you, Alissa. I think we still have time for a few questions. So I see a hand at the back there. We'll get a mic to them.
Yeah, Alissa, thank you. I have a tangential question related to the recent park fire that spread, northward and beyond. And a lot of blue oaks got hit a lot by the fire. But over the years, I've never really seen blue oaks basal sprouting after a fire. So is it just rare, or do they not do it at all?
Yeah, I haven't seen it very much either.
Yeah.
I'd say it's at least rare. I mean, I've worked a lot around black oak, huge basil sprouts after
Yeah,
fire.
huge. Yeah,
Yeah,
up
and
on Green Gate in Upper Park, it's the live oak and the black oak and the other oak up there, Oracle, whatever. They're all basal sprouting, but not the blue oaks.
yeah, I'm really worried about blue oaks that burn in fires. I think they're going to need more help, more active.
Right, because I first looked at this. Wes Dempsey took me on a hike in 2000 because there had been this backfire that got out of control like the year or two before, and we were looking at the same thing, but all the plants. But same thing with the blue oaks. There was just no basal sprouting.
Yeah, I do know the ones that we had burn in fires. One was in the 2019, the Kincaid fire. The seedling actually had enough, it came back up, put up another sprout. So that was really promising to see. But I honestly think we got lucky with that one.
Yeah, thanks.
We have time for one last question. I see one up here in the front.
Hi, thanks. I was just curious if you have a theory as to why there was herbivory on the local seeds versus the xeric acorns.
Yeah, I was like, somebody's going to ask the herbivory question. I think that one, I believe there's a couple different theories. One is that those drought-tolerant oaks are put more energy into developing their defenses against herbivores. It might be one of their adaptations. But that is something that I would contact. Blair's email is on the slide. I would contact her and find out. We have ongoing experiments also at the Hopland Research Station in Mendocino County, and I believe they have started, like they've got a couple master students working on the herbivory question as well.
I was just trying not to jump to conclusions, but I was thinking about like pollinators and local endemic plants. They seem to favor them in some studies. Anyway, thanks.
Thank you.
Thank you, Alissa.